I'd like to make an observation here. It's wonderful to see students, of course, some students sit in front, although it's very rare, but most of them try to sit the back seat. But one thing I've noticed about faculty members, they're always occupying the corner seats. You see, they're all lined up in the corner seats, all have some plans in their mind that the corner seats would enable them fast exit. So, oh, right. Okay. Well, yes. Tough faculty members too. Right. Okay. There'll be some more people coming. I'm just waiting for one more minute. Yeah. As predicted, the corner seat with all faculty members. Look at that. Okay. I'm going to start. So, I welcome everyone for this edition of the Wednesday Colloquium. And sometimes I feel lucky to do this hosting part. Of course, it's a lot of work, but when you get speakers like today, it's always worth it because you get to hear all the history behind the start of a field or, aspects of the fields which you are so fascinated about, but you have a person who probably saw it grow and saw the most important experiments. So, as usual, welcome Professor Amitabha Chattopadhyay from CCMB. And I have been meeting you for some time now, in different conferences, first time in Shillong in FCS meeting. So it's a pleasure to introduce you formally for the first time. So Professor Chattopadhyay actually did his bachelor's in chemistry from Xavier's in Kolkata, not the one in Mumbai, Xavier's in Kolkata. Then he went to do his master's from IIT Kanpur. Uh, and this was, long time back in the early 90s. And then when he decided to do his PhD, he went abroad. He went to Stony Brook, um, which is the State University of New York, which is sunny Stony Brook. And after his PhD and a postdoc at UC Davis, he came back to India. For the long time, Amit's work has really homed in to the role of biological membranes in normal and diseased condition. One of the most important work of his entire lifespan has been to elucidate the role of cholesterol that is bound in the membrane in various aspects of important functional aspects of biology. And he has been using this, of course, he is a biologist from a chemistry aspect. So he has been using a lot of understanding of, interactions, membrane, protein, and, cholesterol protein interactions in understanding functions of things which are as big as a GPCR. So based on his work and especially based on the physical methods to follow interactions, things which can follow dynamics like fluorescence correlation spectroscopy, FRAP, fluorescence recovery after flow to bleaching, all these techniques which allow for measurement, careful measurement of dynamics of biomolecules on membranes, he has been able to parse out really important aspects of how cholesterol is functioning in different, for example, how it infects a host cell. So, but without further ado, I just wanted to bring to notice to everybody that Because of his fantastic work, he's been awarded and rewarded from various places. But one of the important things is he's the fellow of many academies, all national academies and international, the Royal Society as well, the fellow of Royal Society as well, and also the Shanti Swarup Bhatnagar Prize winner. So with that, I welcome Amitabha for the Wednesday colloquium.
Perfect. Yeah. Okay. So thank you, Jyotishman, Debashish, and Sandhya for the invitation. And it's a pleasure coming back to TIFR and seeing a lot of friends, making new friends, and soaking yourself in the energy of TIFR students and faculty, and the ambience, of course. So I'm going to tell you a story today, which central character is cholesterol. This is not a research seminar from my laboratory, as I was told. I do that in other places, and you can hear that. I will have some of my work in this, but this is a story I have built based on membranes, which I work with for a long time. And I'm going to make a context clear. If there's a doubt, you can raise a hand, or you can ask me after the lecture, and just enjoy and ask me if you have any doubt. Sometimes it also helps me. So central character is cholesterol, but there's a context of everything. Cholesterol that I'm going to talk to you about is from the cell membrane particularly. Now, for those of you who are not biologists, biochemists knew about 150 years back that a cell is not a bag full of enzymes. A cell has mini bags inside it that has called mitochondria, nucleus, in a Golgi, and so on and so forth. And they have their own little membranes that allow them to have their own identity. However, their differentiation, their development, their rate of growth are different from the membrane that puts it here. So we work on biological membranes, plasma membrane, which is the outermost one, and also the inner ones. Now, this is a model. So when you talk of DNA, people talk of the Francis Crick and Watson model. This is similar model, of course, very differently arrived by Singer and Nicholson. This model is old because this model is more than 50 years old now, and but still it is the basis. But now we know this model is not enough, although it's still a good structure to understand. You can see bilayer of lipids. This is a protein that is hydrophobic enough to penetrate the membranes. Just to give a perspective, this whole thing from this ball-shaped object to this ball-shaped object is typically 5 nanometer or 50 angstrom. Okay, so that's the scale. A lot of talk, a lot of papers, a lot of narrative these days is nanoscience. This is real nanoscience. We're doing biology on nanoscience level. We will be talking what happens in between and inside the lipid. This is the physical perspective. Okay. So that was 72, but this model has continuously modified. Shitama, who is my student and now postdoc in Purdue, and I wrote a perspective on biological membranes a few years back, and it somewhat looks like this. You see, it's not plain as they saw. It's not like a piece of pastry. It's a curved pastry, a lot of colors, and there's some actin cytoskeleton, which Singer Nicholson never thought. But these are proteins that are below the membrane. But we know now they interact with the membrane in a very functional way. And of course, the lipids, proteins, and carbohydrates, which are only on the outer side. But this is not a static picture. This is a snapshot, right? You don't know how it functions. You don't know what kind of time scale we are talking of. So Sritama and I tried to put it together in next 2 slides. So if you go from only this part of the membrane, the half of the bilayer, it looks like this. For those of you who like chemistry, there's a little bit of chemistry here. So this is 2 acid chains. This is the glycerol backbone with some kind of esterification. This is a phospholipid, not showing cholesterol still. And so if that is 5 nanometer, this is 2.5 nanometer. 2.5 nanometer, we see a lot of gradients of mobility, polarity increases this way. This is bulk water, which means static dielectric constant 80, and mobility. And this has been arrived from a whole bunch of experiments on order parameters from deuterium NMR, ESR, fluorescence spectroscopy, some done by us, and nowadays simulation at all time scales, both coarse grain and all atom. So a membrane is not only non-homogeneous, but is heterogeneous to very small time space scale. That's something I want to keep in your mind. And that's what we have said here. Okay, now I told you this is not a static object, and this gives you some is very complicated. Don't worry about what is written on this. If you cannot read, that's perfectly fine. I'm sorry about that. But these membranes, these lipids move this way, they move this way, they move from one leaflet to other leaflet. All these are in different time scales. And what we call biology, membrane biology, and membrane or membrane-bound proteins do 50% of signaling in your body. So if that doesn't work, 50% of the signaling is not working. You can see well, you can taste well, you can visualize, you can smell well, all done by membrane proteins, mainly GPCRs. You see, Luke will tell you it can be as slow as second or tenths of second and as fast as a picosecond. So it's 12 or 50 or more than that orders of magnitude, all put in this little, 2.5, this bilayer, 5 nanometer bilayer. And it is a combination of that gives the membrane's function. So a simple question, because in TIFR people do physics, chemistry, biology, how do you measure this? Well, surely there's no equipment in the world, wherever you go, that you put the equipment and get membrane dynamics. Depending on what you want to study, you have to choose the equipment. If you want to study lipid flip-flop, that's a very small process, a very, very slow process. If you want to use lipid dynamics, it's a fast process. So depending on that, you have a spectroscopic, either IR spectroscopy for fast processes or Raman sometimes. And for very slow, there are other techniques. NMR is one of them, or even slower techniques you do. So there's one shoe fits all, doesn't fit for membrane. Keep in mind that. Okay. So this is elaboration of the same thing. This is a very biased slide because I've worked on the technique called REES. We call it wavelength selective process. This can give you information on sub-nanometer scales. This is energy transfer, very common technique. Many of you know this. This is FCS, fluorescence correlation spectroscopy. This is FRAP. So these are different kinds of, I'm trying to correlate the membrane phenomenon with time scale so that you have an idea, not an exact idea, but an approximate idea, what I should do when I don't. And not only I'll tell you about myth, I'll tell you about funny stories. Singer and Nicholson, who discovered this great model, and this paper is very famous, and if I am not incorrect, Nicholson is still alive in California. They actually used some technique that is not correct for the right process, and everybody thought that, they didn't do this. Some of their colleagues did it in France, a very smart colleague, I don't take the name. They told membranes are static and published papers in reasonably well-known journals. Now we know they only use the wrong technique, right? So if the common analogy I give to school kids is, if you have a bicycle tire and you give it a rotation, after a certain point, you don't see the individual spokes, right? Because they blur, right? But as it slows down, you see the spokes. So it is the time scale that matters in which you are seeing the time window and the phenomenon that's happening. Same thing in membranes. So all these are done and more and many more. I didn't talk of terahertz or Raman, but just to give a perspective that it's a very dynamic, nano-size object in the cell, very functional. Now I'll come to cholesterol. Now, membrane lipids are phospholipids. I showed you that. Cholesterol is a very unique lipid that occupies a lot of narrative, not only in science or biology or chemistry, but also in popular narrative. You do a Google search of articles in journals like in magazines like India Today or any popular, lot of things on cholesterol, sometimes wrong, most of the times wrong actually, but a lot of articles. So cholesterol evokes narratives. So this is how the molecule looks, looking at a boring here, gray and white and one red. This red is the OH group. But all I'm going to tell you is this: to be a lipid in a membrane, membranes are like oil. There's hydrophobic and there's water outside, water inside. You need to be hydrophobic, but you need to interact with the water. So this is the portion that interacts with water, OH group. All lipids have hydrophilic and hydrophobic. Moiety cholesterol probably has the least size hydrophilic molecule, and the rest is, things that I'll tell you. This is some kind of structure. Now, so this is predominant in eukaryotic membrane and controls organism dynamics. That means any cell that has a nucleus and has different organelles has cholesterol, especially higher eukaryotes. E. coli doesn't have cholesterol, although some bacteria like Mycobacterium needs cholesterol for stability and infection both, but they cannot make cholesterol. But human bodies can, and most higher eukaryotes can, most mammals can. Okay, so it was discovered long time back in 1769 by a man called Poultier Poultier de Sala in France, in human bile and gallstone. So he just isolated this molecule and he gave the name cholesterol for a certain reason, and that was done long time back. But people didn't know what is this molecule, what does it do, and look at the time, 1769. If you look at Indian perspective, is about is much before Sipai Mutiny, 100 years back, Sipai Mutiny or something, right? First independence of war. So when a baby is born in a family, you name the baby. The naming was done by this man, Chevreul, another French chemist, who looks like a character in a Satyajit Ray movie called Gupika and Bhagavan, if you have seen the movie, right? And he named it cholesterol because they call for bile and steroids for solid. Those days, and you'll be having a lot of fun thinking, people didn't know molecular weight, but they knew empirical formula. They did not know molecular formula. So they knew how many carbons, how many nitrogens, how many oxygen, but that could give rise to many structures, right? But so that was a big deal in those times, that I know the empirical formula. And this was given by, this time not a French guy, but a German guy, 1888. But Chevreul was still alive when this was done. So, as somebody jokingly said in the seminar at CCMB, if you do good work, you keep your health well, you never know when the bell rings. So, so he knew that. Okay, so you name the molecule, you know the empirical formula, but that doesn't mean you know the structure of the molecule. You're not talking structure of a protein or anything, structure of a molecule of molecular weight about 386 actually. But that was difficult in those days. And so extracrystalline field eventually come. So the first Nobel Prize in cholesterol was given to this man, Heinrich Wieland, again another German guy, who got Nobel Prize in 1927 or around that time. Okay, and he got Nobel Prize for this work in 1927, right? And so he shared it with one or two more people. It was a big discovery, and people have started believing that cholesterol does something interesting, but things are not clear. But 27 he got Nobel Prize. Okay, so this little story here. This is Heinrich Wieland. I know a person called Felix Wieland, who used to be the former editor of Febslater, a journal where I've been an editor for a long time. Once Felix told me to give a talk, I went to his office. I saw a photo of this guy, and I know this guy. He discovered cholesterol's empirical formula, got Nobel Prize, got a structure after that. He said, yeah, I know him as my grandfather. Okay, so Felix also works on cholesterol, mostly sphingolipids, nice guy. And I said, well, some families are, more blessed than others, as some of us felt. But here is the twist in the tail, right? Picture abhi baki hai. What is the baki part of the picture in this case? The structure was wrong. The structure he did, I show here. Anybody who does some undergraduate biology, biochemistry course, this is not the structure of cholesterol. This is the structure Heinrich Wieland did, and his Nobel Prize was given. In today's narrative, today's India, there'll be RTI, the Nobel Prize will be taken, but nothing happened. He kept his Nobel Prize, nicely, peacefully. Felix Wieland told me nothing happened. But he and others started so you could still get a Nobel Prize and be wrong if you're original. That's my take. So it's good to be original, not good to be. And I keep telling my students, as long as they're original, it's okay. And be honest, of course. The correct structure came in 1932, but J.D. Bernal, many of you know his name, he was a great crystallographer after Bragg. They found a real structure, but Wieland was one of the collaborators. So he didn't think it's us versus them. He said, well, let's try in a different method, see what we get. And they got this structure. So the truth came out. Nobel Prize was in the Wieland family, no problem. That was 1932. Now, I have a student who is no more with me. She's a faculty now. She used to do these timelines, which I love. Just to put a perspective, so this is, starts in 1786. This is all I told you. So Overton is one of the guys who discovered early model of membranes, and he suggested cholesterol in cell membrane, one of the first, And then all these things happen. And now I'll tell about this. How is cholesterol made in your body was not known, even if you know the structure. That was done in 1964 or late 50s, early 60s. And the Nobel Prize was given to Konrad Bloch. I'll tell a little bit about him. And then around 2007, Brian Kobilka and Ray Stevens solved the most important GPCR structure, G protein coupled receptor structure with cholesterol in them. That came out in Science, and they got one of them got Nobel Prize for that. So that's where the timeline ends. So cholesterol has evoked a lot of Nobel Prize, a lot of interesting discoveries. This is just a snapshot of that. Okay, so now let me come to what we know about cholesterol structure and what we don't know about cholesterol structure. At least many don't know. This is the copybook, textbook structure of cholesterol. But the copybook textbook structure doesn't tell you two things. One, I told you OH group is very small. Notice I have color coded the different parts of the structure. Now, many people who do physics or biology sometimes think, oh, chemistry is boring, right? You formula, that's, I grew up with that generation. I have a brother who is a physicist, and my major task is to convince chemistry is very interesting, sometimes even more than other subjects. So the confusion here is, these rings are, how many double bonds are there? One. Now, there's nobody in this room who doesn't know that we know benzene is planar, sp2 hybridization, right? You have read in high school, I read in also high school. And cyclohexane is chair and boat, so it's sp3. This has only one double bond, but a lot of time between late 19th century and late 20th century, early 21st century, people struggled between 1990 and 2008 or something to find out is cholesterol planar or not. And one person who was in this narrative used to live in Bombay. Unfortunately, he's no more. He used to work in IIT Bombay. His name is Professor Anil Lala. Anil Lala used to be in this game, but he actually published very nice papers showing cholesterol is actually quasi-planar. It's almost planar, but not really planar. And they can say, okay, it's an esoteric idea. How does it matter? Nowadays, not only grant givers, but also so-called important people from Delhi and other places tells us, but so how does it matter? It does. I'll be talking about a disease called Smith-Lemli-Opitz Syndrome, which happens because there's one more double bond in that patient. And especially for students, I'll tell you, it has no drug. Why students? Because nowadays people tell me it's very difficult to teach students because you say something, they Google it, and say, oh, it's known, right? So drug is known, why should I study, right? Not all students, but some students do that. First thing I said, there's no drug. I know people also Google something. They say, no, there's no drug. I say, people survive, people live with that. But we'll tell something that will give some hope, all because of one double bond, one excess double bond. So it's the important double bond, right? Okay. Now in 2D, it looks even more interesting. If you look at Indian mythology, you see gods with two faces, four faces. Brahma has four faces apparently. Cholesterol is two faces. One face is a tough face. It's a rugged face. In all methyl groups. This is a smooth face, all hydrogen atoms. So the smooth face aligns with the long tail lipids, and this aligns with the corrugated membrane protein, which is bulky and corrugated. And we have shown by simulation, and I think I have a slide, and others have shown by other methods that this actually happens in real life. Cholesterol not only binds to animal bodies, it binds with specificity towards sideness. Right? I don't like you, I don't sit beside you, that kind of thing. But it's not like you, not human, but it's molecular. So it's about interactions and stuff like that. So cholesterol structure is very important, and touch me not. You cannot touch it. If you touch it, put a methyl group, put a double bond, what chemists call a modification, it doesn't work with cholesterol. It's no more cholesterol. Okay, so so just a fact, just tell you, the human body produces about 1 gram of cholesterol each day, and the total cholesterol level in the body is about 40 grams, because you have homeostasis and stuff like that. 25% of this amount is located in the brain. The brain carries max amount of your cholesterol. I have a friend called Shahid Jamil, who was 1 year senior to me in IIT Kanpur, and Shahid and I joke about each other a lot, some of which I won't tell you now. But first time Shahid heard one of my lectures, he said, UP में बोलता है कि आपका भेजा में चर्बी जम गया. So this is actually correct. I said, yeah, this is actually correct. Most of your, A is in brain, in the cholesterol, both in glia and in neuron. Okay, so this man, Konrad Bloch, was a biochemist, organic chemist and biochemist in Harvard biochemistry department, worked very hard to get cholesterol biosynthesis done. He did the work in 1956, got the Nobel Prize in 1964, and his postdoc, who later came to India in other part of Bombay, IIT Bombay, worked on some of these problems in the early career. Anil was much senior to me, but we used to collaborate. And unfortunately, he passed away. But Anil was one of the early guys to do biology in IIT. And for those of you who don't know, I can tell you, I'm a chemistry student who wanted to do some biology at the inclination in IIT Kanpur. And I was told that I'm too smart to do biology, something like that. Biology you don't do. But that has changed. Many IITs now have good biology. But Anil was one of the early guys who did very good biology in IIT Bombay Biotech Department, which was actually founded by him. Okay. Why am I? So this is Anil, who used to do that work. So I don't want you to read these structures, but I want to tell you that it starts with acetyl-CoA, a small molecule, and there are 29 enzymes after 20 enzymes that work in a very cooperative way to give you a product called cholesterol. You have to trust me, this is cholesterol, this is acetyl-CoA. So this process is very well controlled by feedback loop and things like that. How did this man discover it in 1950s? Around that time I was born, and I've been here for a long time. Well, he radio-labeled individual atoms with carbon, and then went ahead and looked where, it's like looking for a needle in a haystack for each carbon, not that. It's tedious work. I'm not so sure I'd like to do that kind of work, but the result of the work is very good. So that's what Konrad Bloch did, and one of the small things I picked up from an early paper of him, 1926, is he fed squalene, which is the intermediates you see somewhere there, to animals, and that led to an increase in cholesterol content. So midway, you get an intermediate. If you externally feed it to the rat, then your cholesterol levels go up. So that means you are actually up-regulating that particular path. So that kind of thing. Bloch, of course, was a smart guy. He did many other things. One more thing I want to tell you. Many of you know about hypoxia, the condition in which you have less oxygen. So if you go to higher altitudes, you sometimes have breathing problem, problems with your consciousness, and people faint and things like that. That happens because the oxygen levels are low. So there are things that are not working right. Now, cholesterol, so was evolved in Earth because oxygen came to, initially there was no oxygen, then oxygen came. So if oxygen is less, there's no cholesterol. I just write, cholesterol biosynthesis is an oxygen-intensive process and requires 11 molecules of oxygen for each molecule of cholesterol biosynthesis. So if you don't have 11 molecules, let's say you have 2 molecules, you have hypoxia. And hypoxia is a very dangerous thing. Unfortunately, for India, lot of our battlefields up in Kashmir and are all in higher areas. And many of the soldiers who go there are not acclimatized because they are from South India or other places in the mountains. They have huge problem of depression. This is not very commonly publicized because it has to be military and there's some sensitivity, but it's very sad. These people are actually helping the nation, but they are, getting very, so generally they rotate them. But one reason they have it is because of this reason. Lack of oxygen suppression cholesterol bias is giving rise to hypoxia. We did some work on this, so that's a different thing. So Konrad Bloch was a good chemist, I told you, good biochemist. He was also a good biologist, turns out, because he gave a hypothesis much later in his life, in 80s, I think. He said that cholesterol biosynthetic pathway, which I showed you, is a complicated pathway, parallels cholesterol evolution. Let me explain this. Biosynthetic pathway is organic reactions taking place in your body, 20 enzymes. But he said, if you see the product that each reaction is putting, you will get those intermediates in, I told you cholesterol is not there in E. coli, it is not there in yeast, but yeast has ergosterol. It is, but yeast is eukaryotic, but not higher eukaryotic. So slowly as evolution happened, cholesterol structure also got modified. It's a huge statement to say in evolution. And he actually, and so cholesterol precursors should therefore have properties that gradually support cellular function of our organism as they progress in the pathway forward to cholesterol. The idea was, since, as many of you know already, membrane proteins are more in Homo sapiens or humans compared to lower eukaryotes, because this is where cognition and everything is there. So you need, those proteins need some not only structure, but also dynamics. They need to rotate right, they need to translate right. Cholesterol allows that, because cholesterol, depending on how much cholesterol you have in the membrane, allows optimum dynamics. And that's provided by this. Okay. So that's what he said, that if you don't have cholesterol or something else, it will look like cholesterol, but it's not. So its dynamics will change. That will affect life process. Very deep thinking, using some very simple, I would say, experiments. And he published it in somewhat in early 83s. But we think he was very correct. And another Nobel laureate, Brown and Goldstein, who got, many of you know this, they got Nobel Prize in 1985 for discovering LDL and HDL, said that cholesterol is the most highly decorated small molecule in biology. Decorated meaning not the other way, but it's like every atom on that molecule of molecular weight, only 386, not in kilodaltons, is very essential. It's functional. The decoration is not for decoration purpose, but functional purpose. So I told you, I'll tell you about this disease, and I have an interest in this because we have done some work on this. This disease happens because now I'm not showing all the steps. One of the last steps, the penultimate step of cholesterol biosynthesis of the one pathway called Candace-Russell pathway. The only difference, if you look very closely, this molecule and this molecule is this double bond cycled in red. So those days we used to read cartoons in newspapers that find the difference between two photos or two pictures. If you see that, the only difference is this. So people who have, so how are lipids made in your body? They're made by enzymes. These enzymes are coded by genes, but lipids have no genetic background otherwise. Okay, so if the enzyme is for whatever reason mutated, right, at any one or many places, now we know more than 150 mutations have been identified, then you don't do the reduction the enzyme is 7-DHCR. So this is a reductase. It reduces 7th carbon to double bond to single bond. That is reduction, right? You take out a and then depending on the severity of the mutation, the ratio of cholesterol to this will be changing. In normal human beings, many people in this auditorium, you cannot really measure this very well, even with today's lipidomics techniques. But once you have cholesterol, SLOS, you have some amount of this. And cholesterol, of course, you can always measure. You make the ratio, and that's how doctors decide how severe is your SLOS. If it is very severe, the child is born dead. If it is Slightly less severe than that, you live by 9 years and then you die of cognition or something, or problem. Maximum, you can live up to 30, but you cannot live independent life because you cannot, you do not have the capacity of mind to have independent life, all because of one whole bond. So this is the disease discovered by 3 physicians, Smith-Lemli-Opitz in 1963, but the molecular cue came in 1994 in American Genetics Journal. So how do we handle with this? So here comes a myth. If you look at literature, public literature, eating eggs, non-veg food, these, that, bad, a lot of cholesterol is bad for you, right? This is a disease of less cholesterol. You die, right? You don't have cognition. If you have more cholesterol, there are drugs. You can still live depending on your genotype, and there are ways to handle this. But low cholesterol, nobody talks about it. Low cholesterol, you're depressed and you're gone. Okay. In fact, I have a friend called Sanjeev Jain in NIMHANS, who is a neuropsychiatrist. And NIMHANS, for many of you who don't know, is the largest mental hospital in Asia. Sanjeev, when I told Sanjeev this, he said, well, I don't know all this molecular stuff, but the simple thing we used to do to depression patients is drink milk with cream with dark chocolate at night. It's nice, it sounds like good. I'm not depressed, I don't mind taking it. He said, no, but it really works. So that makes sense. So the myth is this: only high cholesterol is detrimental for human health. That's not true. Cholesterol should be in optimum level. Low cholesterol is equally bad, actually more bad than that. But has somebody told you that? No. That's one thing. Well, there are some indication of that nowadays. There have been some celebrities who had bulimia or who died because of, for some professional reason, they have maintained a very unique BMI and body weight on that. But they were often depressed. Many of the popular stars, some of them very famous now, have acknowledged that they tried committing suicide, this, that. It's all because of this. Because you cut cholesterol, and there are all kinds of mental things, including depression, and suicidal tendency happens. So that's one level of. Yep. So this is the disease. It's a lethal autosomal recessive congenital malfunction. It's not very prevalent, but quite prevalent.
Can I ask you a quick question? There's, the isoform which has the double bond versus not the isoform, the molecule which has the double bond. Yeah. As opposed to cholesterol. What is the phenotype exactly? Is it due to the loss of cholesterol, or is it due to the presence of that precursor, which is...
You are ahead of me. But since you asked the question, I will tell you this. The community is vertically divided on this, not horizontally. So there are equal number of papers, almost, who says it is lack of cholesterol, and others is more of 7-DHC. We recently did some cellular experiments, not animal level, which shows 7-DHC can, add more 7-DHC can do this. But it's cellular level, so you have to be careful. So it's not very known. Some of us believe it could be both, right? This is not very, lot of papers try to do this, but the conclusion is sometimes very biased. As of now, both are risk factors. And interestingly, you expect when 7-DHC is there, cholesterol will decrease. That doesn't happen sometimes. Cholesterol can still, if you assay it, you see it's more or less same level, but there's 7-DHC, which initially is very difficult to accept. But it happens because there are other ways to keep cholesterol constant.
The 7-DHC is also on the plasma membrane, or is it also on endomembranes?
Say it again.
Is the 7-DHC only on the plasma membrane, or is it also on the endomembrane?
Yeah, good question. So, very good question. So, cholesterol and 7-DHC both are also on plasma membrane, also on other membranes. But remember, sphingolipids and cholesterol, 90% of them are in the plasma membrane. That has been proved. 7-DHC, I don't think anybody has looked into this, but probably it's also 90% of the membrane. But what doctors do when they do this test, they don't isolate your membrane membrane, it's a plasma membrane, they do analysis on the serum, right? But by and large, it works out because it's 90%. That's what. But very few doctors isolate membranes, and we do that in the lab. Others don't do it. Second point, there's only one paper from a German group which showed that plasma membrane cholesterol and serum cholesterol is related. They gave some animal, rats statin, and showed the difference in membrane cholesterol and serum cholesterol. And they are not same, but they follow the same track. It's not a linear relation, but so that's all we know about it. But most of the literature about SLOS and other medical journals, you'll find only serum cholesterol. So there's the assumption here we are making that since 90% is plasma membrane is okay to work. But it is not, that's about it. Okay, so clinically diagnosed by this, by Bush. And by the way, 8-DHC is unstable. So you can't keep it for like, it's an isomerization product, basis display structure and brain abnormality. Now, some areas of the world, the rate is more than that. And this is Scandinavia. And that's because the sunlight is less there. And if you look, no cholesterol pathway, after cholesterol is formed, there's steroid, there's a vitamin D pathway there. So many of us Indians, after about 10 or 15 years back, all doctors started telling we have less vitamin D because generally in this kind of rooms, so many of us take vitamin D tablets. But sometimes that also affects cholesterol. So Scandinavia has a very high risk of this, and that's something I should mention. Okay, so these are typical people you have. So this is, for those of you who know development biology, it's a hedgehog pathway, right? It's a early development pathway. Cholesterol is a big thing there because there's a protein called hedgehog protein that actually controls many of these. So you have pattern formation defects. You have 6 fingers or joint fingers or other parts of body shown here, cleft palate. But just because you have this symbol doesn't mean you have SLOS. So doctors, when they have this kind of patients, eventually check 7-DHC over cholesterol, and only a few percentage becomes 7-DHC. Others are, they are cleft palate, nothing can be done. They are not SLOS patients. So this is the phenotype. And so we did develop a long time back a cellular model. We don't have the mutant receptor, but we can use a inhibitor called AY9944 that actually does more or less the same thing shown here. AY9944 actually inhibits this pathway, and by using it in a dose-dependent manner, my student Yamuna, who now works for Moderna, the company that makes the RNA vaccine, showed that if you titrate the cholesterol to 7-DHC and you see some function, like in that case we saw ligand binding, we also see signaling later, we showed that yes, you can reproduce this, and actually this mimics the. Now, how do you know it is actually because of cholesterol? So this we did. I hope I have that. Okay, so I don't have the data here, but if you put back cholesterol, actually you can get back. So because cholesterol does many things. How can you correlate 1-to-1? It could be causal also, right? But so most people have difficulty replacing cholesterol because it's not easy as taking it out. There are lots of tricks, experimental tricks, not conceptual. Okay, but we have done that in this case. But this is old story. My present student, Ashwini, did a nice experiment, and I'll cut it short because I have to finish my talk and then do a little bit more things. Nowadays, we work on GPCR endocytosis, G protein coupled receptors. I'll tell you about that. And we see how it goes into the membrane through clathrin or caveolin-mediated pathway, and how, if you deplete cholesterol in the membrane, it changes. In this context, Ashwini looked into this SLOS kind of cells, where we made the cells having 7-DHC by using AY9944. And we saw how, this is Ashwin, we saw that in this, generally, for serotonin 1 receptor HEK cells, it goes through clathrin-mediated pathway and gets recycled back. Right? So you use tags like RAB11, RAB4, and you co-localize quantitatively, and usually it comes back. When I say comes back, predominantly. Biology, there is nothing called 100%, predominantly. or it can go and get chewed up in lysosome, which is the acidic compartment. But in this case, we saw that even without the ligand, so which means we're not doing ligand endosynthesis, the receptor goes to lysosomes and get chewed up. And this is Ashwini's work published in Traffic. And that was very strange because the receptor should not go to lysosome when you have done nothing to it. So there's some problem with this phenotype, and this has been hinted by others, but we actually showed that. Very recently, we did some work, and it has a TIFR component. That's why I should tell you that Anand Patel is an ex-student of the NMR group of TIFR, and he is a CCMB colleague of mine. We have done some experiments. We did it some time back. We need to write it up now, where we took, what happened. The funny thing about literature, especially research literature, is it is a disease that affects brain, but most clinicians work with skin membranes because they're easy to work with. Now, skin membrane, even if you do all kinds of biochemical and biophysical work, will not tell you about cognition, right? Professor Satyamurthy, who used to be director of Aisar Mohal, used to teach us at IIT Kanpur, and he told us a story of a guy. He was looking for a 10 rupee note below a lamppost. Somebody said, "Did you lose it here?" He said, "No, I lost it where it's dark. I can see, so I'm seeing here." So, scientist always does these things, right? We measure things that we can measure, try to justify it, right? So, so we tried to do something here, and this was an animal experiment. So we took help from Anant and other person to Animal House, and my lab, Jaffrey. So we showed that if you actually treat rats with this and do dose dependence and all that, I'm avoiding that, we actually see, you can quantitate the metabolites, the neuronal metabolites, and we can clearly see some of the metabolites like glutamate, GABA, aspartate. So the pathways that signaling gets affected, you can see a drop in reduction. I think this is a very nice work which actually can pave further work on actually what is happening. But this is just one slide that I'll show and not talk about it. I'll end my talk in the next 10 minutes because my time will be up by saying something we work on GPCRs, G protein coupled receptors. Now, GPCRs are very interesting molecules. They're basically cellular nanomachines. You can think of it that way. That does a lot of signaling, and they are in the membranes. They're nanomachines. Why are we interested in this? Because they transverse membrane not once, not twice, 7 times. So the transverse membrane 7 times. That means the carboxy and the amino terminal. The amino and the carboxy terminal are on 2 different sides. One is outside the, one is extracellular side, and one is intracellular side. So we thought long time back that this kind of lipid protein should interact with the membranes, because we know ion channels does. There are only 4 helices. We know glycophodin does. There is only 1 helix. There's a whole bunch of literature on this. In my postdoc in UC Davis years back, we showed that nicotinic acetylcholine receptor from Torpedo californica, because those days there's no cloning of SHR, cannot function without cholesterol in reconstituted liposomes. We did both patch clamping and ligand binding. But when I started looking for GPCRs, we said nobody has worked on these things. So I thought maybe somebody has done, it doesn't work. So when you're in trouble, you do an experiment. We did the experiment and we showed, I have 10 minutes, I'll finish. Yeah. Did the experiment and we showed, yes, that was done by Thomas Pocadil, who is in Pune, not too far from here. I'm not going to tell you about those, but these are receptors that are very important drug targets, because all it happens is G proteins are, there are these trimeric G proteins that happen in higher mammals, higher eukaryotes, and they couple with, the protein called G protein coupling, and they have ATP bound and ADP bound, and signaling happens when you activate them, the G protein coupling by ligand. This signaling that happens, many types of signaling that happens, actually are targets for drugs. So GPCRs are good drug targets. Strictly speaking, the GPCR signaling that are good drug targets. And in today's world, about 40% of today's drug targets are GPCRs. As of today, it could be 60% within 5 years, because now we know GPCRs that are not in the plasma membrane, but that being endocytosed is also signaling, but different kind of signaling. And a lot of work, including 2 companies, are looking for drugs from those kinds of signaling. So it's very important. Okay, now why am I talking about it, GPCRs? Because I want to tell you something that I've done about it. This is FDA approved 35%. And again, GPCRs are discovered in 1970, 72 by Bob Lefkowitz and Kobilka, I'll tell you that. But they are used as drugs targets even before they knew they are GPCRs. So cardiologists, for example, have been using medication based on GPCR even before GPCRs were discovered or identified. Now, so our story is this. This is Bob Lefkowitz. This is Govind Khorana. And most of you know he got a Nobel Prize because of his work on synthetic gene, nucleic acids. After he got Nobel Prize in 1969, from 70 he started working on membrane protein lipid interaction with some nice Indian and American postdocs. And he was working on rhodopsin, initial vector rhodopsin, then rhodopsin. He did a lot of work on it, and Bob Lefkowitz did at Duke and other. He was an MD and he was a organic chemist and biochemist. So the approach was very different. Personalities are very different, but they eventually came to the same point. And he got Nobel Prize for it in chemistry, 2012. And he joked that I never went to chemistry class. I'm a doctor, I'm MD. He got with Brian Kobilka. So we looked up these things and we saw that most people do at that point till 2000 signaling or pharmacological characterization. So I said these questions. We said, this goes through membranes 7 times. You should interact with membranes. And at that point, we asked 2 questions. Which class of lipids interact with GPCRs? What is their function? Is it known? Or is it same for all GPCRs? Or changes from GPCR to GPCR? And what happens to human health in the biosynthesis of such GPCRs defective? So Thomas Pucadil, now in Pune, showed 20 years back that at least for serotonin 1 receptor, we need cholesterol for function. It is ligand binding, G protein coupling, signaling, or oligomerization, and was done by success students. And I'm not showing you the data. Clearly, this is the receptor for which this kind of lipid started. Eventually, Brian Kobilka and others also started working on beta-hydrogenic. We also have one paper on that. But there are many receptors now which are dependent on cholesterol. So we have shown these 2 receptors. T2 for beta test receptors. They are receptors that signaling depends on cholesterol. But according to current estimates, about 70 or 75 GPCRs are believed to exhibit cholesterol dependent signaling. How many GPCRs are in your body? About 800 plus. So we know 75. Other experiment hasn't done. But just when we are getting very comfortable, papers are getting accepted, we thought, and our reviewers are saying, so what's the new thing? We saw reports that GPCRs has a secretin, cannabinoid, CB2, neurotensin, which looks just like class A GPCRs like ours. They even have cholesterol binding sites, do not change signaling, whether do up cholesterol or down cholesterol. Nothing happens to them, right? So those reviewers who started telling us now, so what's your next project? They stopped, and we did not know why. It's $1 million question. Why these GPCRs are very sensitive to cholesterol, others are not. So a little bit of structural biology. Membrane proteins are difficult to isolate, more difficult to crystallize, simply because they move a lot. And GPCRs move maximum of membrane proteins because they have loops. First GPCR structure came in 2000, and then many structures have come till now. So we did a meta-analysis in 2 years back. We showed that of the structures, about 30 to 40% GPCRs of high-resolution structure, meaning mainly cryo-EM and some X-ray, shows bound cholesterol. Are they functional? We don't know. We also did analysis how. So at that time, cryo-EM was coming up, but now there's more cryo-EM structures. There could be between 1 and 10 GPC cholesterol molecule bound to them. And it so happened that in our receptor, we found out it is about, in a 10, the structure CA21 come in 2021, and the structure showed in Nature paper that there's 10 cholesterol bound. So the fact that many GPCRs are crystallized with bound cholesterol is kind of hallmark now. But having said that, whether these are responsible for the functional things we are seeing, remember this is done in very non-physiological condition. Either you cool the cryo-EM, you cool the molecule to a very non-physiological temperature, or in X-ray, you do lipidic cubic phase. Our membranes are bilayer, they are not lipidic cubic phase. But having said that, that's the best one has today.
Can I ask a quick question? I know you are finishing up. Maybe you can take it after the talk.
Okay.
But is there a specific mode of binding?
I'm coming to that. Yeah. I'm coming to that. So to answer your question, we started looking, is there specific mode of binding? Are there surfaces of GPCRs are big? How does a cholesterol molecule, small molecule, go to a huge GPCR and I'll bind here? We assumed it will bind to the membrane protein, the membrane part. So we saw there's a motif called crack, which could be tyrosine in the middle, amino acids, and then arginine or lysine or leucine or valine. People have discovered crack with other proteins like caveolin 1 that it binds cholesterol. But there's no report of crack in GPCR till then. So crack can be as long as 13, as short as 5, depending on value of X, right? So we showed at that point that yes, GPCRs can have crack. These are the 3 GPCRs with some kind of, in cholesterol sensitivity. And in our GPCR, there are 3 cracks, 2, 5, and 7. Now the question is, this is the problem and challenge with bioinformatics. It can tell you possibility, but you do not know whether this is because of that. So you get lots of things to think about, but are we sure this is the reason? So how to prove it? That these cracks are responsible, because just a crack itself doesn't mean cholesterol binds. So we did 2 approaches at that point. This is Durba Sengupta from National Chemical Laboratory, Pune. Durba and I decided to collaborate that time, and this is the time, about 2012, 2011, when you could not atomistically simulate the whole GPCR, water molecule, sodium ions. It was very difficult unless you have Anton, which one guy had in Stanford. We didn't have that. But you could do coarse grain modeling, which Durba was an expert that time. And so you lose spatial resolution, but you gain temporal resolution. So we wanted to see, can we do microsecond simulation? And we did that. And I'll only tell you a little bit of that here. We have a lot of papers with Durba, but the basic story is this. We found that they are not very tight binding. They bind and unbind all the time. Microsecond timescale, dynamic, weak interaction, but very crucial, shown here. So the energy level diagram is not very deep. It's room temperature. So a small change in pH, a small change in calcium can change this. It's not a very deep minima. And with help from, again, a TIFR NCBS story, with Professor Panikkar and the student Shishupal Singh, then we mutated some receptor. There's a lot of discussion in our lab because if we're a Japanese group with lots of postdocs, we'll mutate everything in crack. But we cannot do that in India, right? So we mutated some, and we all think we know chemistry, so there's a lot of debate in the lab. Eventually, we did only helix 2 and 5. 7, if you mutate, all kinds of things that happen through signaling, we know, so we left 7 like that. And we could not have done it with Shishupal, with advice from Panikkar. So we did all these single and double mutations. To cut a long story short, what are we seeing? We are seeing, so serotonin 1A in hippocampus, which is the part of your brain which generates and stores energy. So if there's a damage in your hippocampus because of an auto accident or a bike accident, you lose memory, either partially or fully. You may get it back, you may not get it back, depending on the lesion, right? So there it is coupled to GI, that is inhibitory G protein, which means upon activation by ligands, is decreases cyclic AMP. That's our assay. This assay is based on energy transfer with some fluorescence that I'm not getting into. So Parijat and Aditya, now postdocs in Stanford and Michigan, did the experiment. It's a complicated slide, but it's not very difficult to follow. So the wild type, if you deplete cholesterol, doesn't show signaling. It's a reduction in cyclic AMP. This is the type. Others also more or less behave the same. We did all these mutations, but nothing happened. So we're not very happy. And we are not mutation kind of group. But 2 of the mutants, which has lysine 101, didn't show any difference. You see difference, but it's not statistically significant. So lysine 101 senses cholesterol somewhere or the other. So that was our conclusion. We published the paper that time, which was recommended for faculty of 1,000. But we also could do some atomistic simulation, and we showed that it actually binds. And here you see, this is something you can do with simulation, cannot do with experiments. You can actually do snapshots of this in reasonable time. And you see that this taking, again, is microsecond, but the difference between this and this is the methyl groups are this way and methyl groups are here this way. So just the little flipping takes 0.7 microsecond. Jana Salent is an excellent person in Barcelona who collaborated this time with all atom simulation. And we also should. Now we're now working on, so GPCR endocytes, the way we look at it is spatiotemporal regulation of GPCRs. Too many on the plasma membrane, too much signaling, put somebody down. Right? It's a common biological response. And we have done a lot of work on this in the last few years. And it's a very interesting field. And as of now, not very crowded field. So the last slide, last slide, then I'll acknowledge, is fantastic result. If you deplete cholesterol from the membrane, with time, endocytes is completely inhibited, right? All you are doing is depletion cholesterol. You are no mutant, nothing. It is just a wild type receptor. So this was surprising, but not that surprising, because there are reports of some receptors which apparently cholesterol forms these coated pits, the curvature. But when we took the mutant of K1, that could, so that restores endocytes. If it's a physical process, one mutation in the 423 amino acid protein will not do that. So we're still thinking what is the mechanism, but the implications are huge, and I'm not good. So these are some of the people who did the work, students, postdocs, collaborators. GK is there. We used to work with GK at some point. And of course, money is needed. This is Salome from Toulouse. We did, so one other thing we did, and Jyotishma and I talked about this, we wanted to do actual experiment on receptor dynamics in 3 time scales, FRAP, FCS, and single particle tracking. And believe me, even if you have the same construct, same cells, you don't get the same diffusion question. And there are reasons for that from which you will learn. Single particle tracking, we didn't have the setup. We wrote CEPIFRA grant and did with Lawrence Salome. And this is, a lot of people helped us in this, and we won't be there without it. Work was done in CCMB. This is the Hussain Sagar Lake with statue of Buddha. And last but not the least, this is the journal where I've been editor for more than 1 year now, chief editor. This is a very classic journal of membrane biology. If you go back, you will see lots of fantastic concepts of membrane domains, rafts. Originally has been published in this journal. You don't need to publish with money if you don't want to. And all our editors are active scientists. So if you have a paper, please send us. Thank you very much, and I'll be happy to answer questions.
Thank you, Amitabha. Questions? And there is already a recommendation with your story with dark chocolates. I recommended my dean to get some snacks with dark chocolates. So anyway, questions?
Yeah, just speak in the mic.
Okay.
While I drink water.
Are there now no questions? Then I can ask. Okay, there's one.
So as you mentioned, that more than 90% of the cholesterol is a part of the plasma membrane in a cell, especially
I
in the...
hear you very well, yeah.
So you mentioned that more than 90% of the cholesterol is a part of the,
the
plasma membrane. So other than affecting the activity of these GPCR proteins, does it directly play a role in determining the neuronal signaling or the process of excitosis? Does it hamper it or any effect on it?
So the first part of your question is the 90% is correct. That's not our work.
Yeah.
Somebody else's work.
Yeah.
What's the second part?
Does it have any direct impact other than impacting the GPCR proteins? Does it have any direct role in determining the rate of exocytosis or the signaling process?
Could you tell me the question?
Cholesterol has an impact on the rate of exocytosis.
Rate of exocytosis.
Okay.
Does cholesterol have any effect in the rate of exocytosis?
Huh.
Does cholesterol have any effect?
Oh, rate. No, we haven't done any experiment on that. But there are experiments done on one other receptor by Fred Maxwell's group. They show, yes, it does change the rate of not endocytosis, but the recycling back. But that's a different receptor, not this receptor. So we haven't done the experiment, so we won't be able to tell you. What we can tell you, though, that if we use not the wild type, but K101 mutant, I didn't show you the data. That's under, we are doing more work and trying to publish it quickly. Not only endocytosis stops, but it gets slowed down. Okay, so we actually chase it for a longer time. Slows down by significant time, in tens of minutes. We don't know why. So there is a change of rate, probably more at the later part of the endocytosis. So something similar to what they saw, but we haven't really quantitated the result as well as we should. So I will not talk about it much, but the literature evidence it does happen with other transferrin receptor. I'm not getting the name. Transferrin receptor, it's shown by Team Magro's group. Yeah, but good question. Because kinetics may change. And the interesting thing in biology is if you wait longer, everything will happen. So the time scale matters, the time window. So your question is extremely relevant in that context, but we really haven't done as much. Since our endocytosis conditions are fixed, we can only talk about that feature. But we did some work later, which we are continuing. We did see slowing down, and we think that's the reason. Good case. Thank you.
Okay. I have a question. Oh,
yeah.
there is a question. Okay.
So what is known about the difference in cholesterol concentrations in the plasma membrane versus the endosomes, lysosomes, etc., and how does that affect
plasma membrane versus the inner membranes?
Inner membranes.
This is interesting question because, so the question is for those of you who didn't hear, what is the relationship between plasma membrane cholesterol, which I said is 90%, and the other organelles below? So cholesterol is made in your body in endoplasmic reticulum. All the 20 enzymes I showed are endoplasmic reticulum bound membranes, bound proteins. But just the richest man lives the poorest life. Endoplasmic reticulum keeps minimum amount of cholesterol or organelles. The rest of the cholesterol comes by vesicular traffic to plasma membrane. There has been a lot of hypotheses and some experiments done on this. One of the experimental hypotheses that I like to believe is, cholesterol thickens membrane bile layers. We know that you can do it by diffraction. So plasma membrane is typically the hydrophobic thickness is typically 30 angstrom, 32 angstrom, depending on what lipids. Golgi is much thinner than that, and nucleus membrane is even thinner. The other way to look at it, and there's a Cell paper by Bianfredi's guy on this, if you take an average ensemble of membrane proteins in Golgi and one in plasma membrane, Golgi proteins have 15 amino acids, so membrane is not that thick, and plasma membrane is 22 amino acids. Remember, if these are alpha helices, each amino acid means 1.5 angstrom rise, right? That's from very basic structural biology. So it appears that cholesterol has a gradient. I have a slide for it, but not here, of increasing toward the plasma membrane. And it has physicochemical and biological implication of that. And this is maintained because there's also retroflow. Now, the controversy, not a controversy, but the discussion about that 10 years back was, is it vesicular traffic or some other traffic? But Jim Rothman and others clearly showed, and is generally believed now, it's vesicular traffic that takes it. It's a very interesting thing. And if you're interested, then there's a very nice commentary in Cell in 1993 by a man from Cambridge. His name is, I'm forgetting his name. It's a 2-page. He gives, and he thinks it is the way to sort proteins that goes to plasma membrane or Golgi or something. One is signal sequence hypothesis that's already there by Global, but this the physical intervention also works.
Can I ask quickly one more before? So that SLOC disease, how do the doctors diagnose? I mean, that double bond difference. Is it like, what is the diagnosis?
70%.
Yeah. How do they diagnose that the difference between cholesterol and the one with the double bond?
You basically measure amount of 7-DHC and cholesterol and make a ratio. So there
like
are some
TLC kind of experiment or thin layer chromatography, or what do they do?
They do some kind of lipidomics, GCMS kind of.
GC mass spec. Okay.
And there is a standard range of normal people. It's not one number. No, if you're below that, you have SLOS, and depending on how below you are, you have three kinds of SLOS: 1, 2, 3.
There's a question.
It is done in India. Karnataka, Telangana has SLOS patients. Koti. Yeah.
Hi, Amit. Excellent cholesterol biology. So what are the other proteins other than GPCRs on which cholesterol has actually functional significance?
Many. I said ion channels.
Ion channels.
There's a huge literature on ion channels. Remember, Pancho Barantes used to come he used to be a frequent visitor to TIFR. He's in Argentina. He has worked all his life on effect of cholesterol and nicotinic acetylcholine receptor. Potassium channels, somebody works the voltage gated potassium channel. Many ion channels behavior depends on cholesterol. That's known. There's big groups working on it. There are proteins like calcium ATPase, which is classic example. You go to membrane biology book, they share if you change cholesterol concentration, how the function changes. So there are plenty. We found it's very strange that they haven't looked at GPCR. Strange for them, good for us, right? And we then thought actually 1 or 2 groups had noticed it, but because they didn't understand it, they put in supplementary or something. Didn't write anything about it. So
very
published the paper, we thank.
One other small question is that, so in addition to alpha helical proteins, whether GPCR or non-GPCRs like ion channels, so cholesterol, does it affect on beta sheet membrane proteins as well?
Yes, it does. It does. It does affect the protein kind of, but it's not that drastic as far as I know. But I think Mahalakshmi, R. Mahalakshmi, in ISAR Bhopal is trying to do something with that. Okay. And so one reason the work has been done with beta-barrel proteins is beta-barrel proteins are predominantly found initially, at least in E. coli, others, they don't have cholesterol, right?
Correct.
While these alpha helix proteins are signaling proteins, right? Because a lot of signaling happens through alpha helix. That's the thing. But I cannot say beta-barrel proteins don't get, I don't read the literature as much, I have to admit. So I'm not the best person to answer this question.
Yeah,
I think there are some work they have done on this. Also, Lucas Stam did something long time back.
Yeah.
But the majority is still alpha helix proteins.
Alpha helix, because
it doesn't have to be 7 transmembrane.
Yeah.
It could be 4, 2, anything. Yeah.
Because the surface pattern of the charges or the residues slightly different on the beta sheet proteins versus alpha helical bundle.
Right. So the surface area changes.
Area.
Okay. Can I ask a last question before we move in for snacks? You said lysine was the critical residue that you identified looking at these GPCR. So I had this question whether things were adventitious that while crystallization you are measuring them, the binding of the cholesterol. But then you said that, you have this with Durba, you figured out that there are some binding modes. However, I have 2 parts to the question. What is the binding constant of such situations of these cholesterol binding pockets? And also second, how does the lysine interact with the cholesterol? Is there some interaction?
Very nice question. Yeah. So I'll answer the second question first. So I didn't show you in that paper, Science Advance, we showed that, or Yana Selen's results show that lysine interacts with K101, both in the crystal structure that came out that year only in the Nature paper, and it also showed in our very high resolution simulation. It interacts electrostatically. Okay.
With the OH of the cholesterol.
So lysine 101 and cholesterol. So cholesterol has so it's a pi kind of it. Pi negative.
The one double bond that you are looking
at. Right.
So you're saying the olefin and the...
Yes. And we showed that in the paper. Second thing, the binding constant. This we discussed with Durba a lot initially. So we don't want to say binding constant. We say occupancy probability. Because the binding is so labile, binding constant can mislead things. Okay. So they are really occupancy, and we devised a quantity, a parameter to quantitate that, and that was maximum occupancy. If your simulation is 3 microsecond, and and you look at residue by residue, at some residue cholesterol is there for 100% of 3 microsecond, it's 1. So elective way to do this. We like to not discourage, but we like to not to encourage people to think like binding constant, because binding constant, thermodynamic binding constant, will have other characteristics that we don't see here. Okay, so it's a very
It's a transient effect.
it's a transient occupancy, but as I said, transient but functional. So whenever so classical biochemistry told you that affinity of that binding changes with biology, or biology changes with that. We are trying to say that it's not that. So it's slightly modified thing, right?
Well, Amitabha, thank you very much for this illuminating colloquium, and thank you for carrying through the entire talk. So I would like all of us to come together with Amitabha for snacks, and he is here for the next 2 days. So he's a fantastic resource person in both spectroscopy, imaging of membranes, as well as about GPCRs. So please make your time and talk to him. Okay, thank you.
Thank you. Marjira.
Very nice.
General audience, no?
History of the
Colloquium definition,
Yes, absolutely.
Very nice.
Thank you.